Hi everyone. I wanted to do a quick little sort of update on where you should be with regard to your two practical exercises. Now, at this point, some of you may be ahead on this if you incubated your organisms over the weekend. But at this point, you should be here, 4 tubes, each with one culture in each tube. They should be slightly cloudy. If yours are not, then just jiggle them around a little bit and you should see some cloudiness in there. We call that turbidity, and that allows you to know that you have growing bacteria in those tubes. Now, if you've gotten to that point and everything is incubated, ideally 24 to 48 hours. 24, depending on the conditions, warm and moist, warm and dark, depending on how warm, you may very well have seen growth after 24 hours. We always say 24 to 48 hours. If you don't see it in 24, you definitely should see it in 48. Okay. So your next step then is to proceed that's just the preliminary. Your next step is to proceed with the exercises, the streak plate isolation, and the Kirby-Bauer antimicrobial disc assay. Here on both of these, you're going to be using your agar dishes. Um, these little plastic dishes, and there's a layer of translucent material in there. That's agar agar. It's just a seaweed-based medium used it's just a general medium used to grow bacteria. You're going to end up using 3 of these plates, right? One for the streak plate isolation and one each for the Kirby-Bauer, right? The Kirby-Bauer, you're going to do one of these plates. You're going to inoculate with E. coli, and the other plate you're going to inoculate with Staphylococcus epidermidis, and then you put your antibiotic discs in there, and then you incubate all 3 together, incubate them all together. So what I've done here, I've just cordoned off my plate. Now this anything you like, if you want to make lines like this or notations like this, make sure you do it on the bottom. That's the agar agar side, okay? The top, because the top, right, is loose and it can move around, so you don't want to write it. You want to write. You can write your um, organism, E. coli, on there, and you can write the date, for instance. But anything that's pertinent to the experiment, you want to write on the bottom. All right, so remember that you're going to mix the two. You have a clear plastic tube with nothing in it. In that tube, you're going to mix the 2 organisms together, right? And then you're going to take one of your little inoculating loops here and you dip it into the mixture. And we call it a streak plate because you're going to streak on the disc. So you're going to pick one of these quadrants and starting at the top, and I have a little video to explain this better, but starting at the top, you're going to streak down, and then you use a new fresh loop, and you start at the point and you streak up into the next quadrant, and then you use a fresh loop and you grab a little bit from that top corner and you streak down to the center again. And then finally you use another fresh loop, clean loop, and you grab a little bit of that point, right, and you streak up into the 4th quadrant. Then you incubate it, and what you should see in the 1st quadrant, a lot of growth. In the 2nd quadrant, the growth has thinned out a little bit. In the 3rd quadrant, you should see clearly 2 different colonies. One is going to be cream colored, the other is going to be red, And in the fourth quadrant, you will see a separation, cream colored and red. And so then what you would do as a microbiologist trying to figure out what these organisms are, you would take a fresh loop, you would touch, for instance, the red colony, and then you would streak it on a new plate or an agar slant or an agar broth. What you're doing is you're now growing a pure colony of that red, and then you would take a fresh loop and touch the cream colored colony and inoculate some other medium to grow a pure colony of the cream. And then you can do gram stain and differential staining and spore stain. You can do all those other tests to determine what the organisms that you have are. Of course, we know because we're just doing this as an experiment. Okay, so for your streak plate, you mix the two organisms together, and you're going to use 4 of these to conduct the streak plate. What I usually do is put a piece of tape around 2 sides just so it doesn't pop open. And then when you incubate these, you incubate them upside down because the moisture will condense. If you do it this way, the moisture condenses on the top and then it drips down onto the organism and that contaminates. So you want to make sure you incubate these upside down. And again, you're going to incubate in a warm, dark environment, just like you did with your 4 tubes, okay? Warm, dark environment. Check it after 24 hours, check it after 48 hours, and you should see lots of growth there. Now for the Kirby Bauer, that's the antibiotic disc, that's the little paper disc, you should have 6 of them. 2 have nothing on them, those are control discs, and the other ones have TE, that's tetracycline, and B, that's bacitracin. So you're going to use a tetracycline disc, a bacitracin disc, and a control disc on each of the 2 plates, one for E. coli, one for epidermidis. You're going to use a fresh, sterile cotton swab. You dip it into the culture tube, and then you completely cover the agar, surface, okay? And then once you do that, one with epidermidis, one with E. coli, once you do that, then you can place your 3 discs equidistant apart. Make sure that you write on here which is which. Make sure you write on here um, S. epidermidis on one and E. coli on the other, okay? So you'll be able to tell them apart. Once again, you're going to incubate. I put a tape around 2 sides. You're going to incubate them upside down, incubate them with your streak plate in a warm, dark environment for 24 hours. 48 hours, you definitely should see some growth there. And then you can do your measurements of your zones of inhibition and complete the data sheet. All right, I will post 2 helpful, I think, videos in class resources to help you out with getting started on the streak plate and the Kirby Bauer. If you have any questions, concerns, message 804-229-7993. Use your Halo inbox, and of course, GCU email is good to get a hold of me. All right, have fun with that, and keep me posted. Let me know what's going on.